From Microbe TV, this is Office Hours for Wednesday, June 26, 2024. I'm Vincent Racaniello, and welcome to our little viral corner of the internet. Yeah, it's been a long time. It's been like 6 weeks, but we're back on track now. And I'm happy to see all of you here. I figured no one would come anymore because it's been so long. People forget things and then that's the end of it. But we'll work on getting everyone back because viruses are always a thing. I want to welcome our moderators for tonight. We have Steph from San Francisco, we have Les from California, we have Tom from the Oregon Coast Range. And let us know where you're from, folks. I'll give you a shout out and welcome you to this rebooting of Office Hours because it's been a while, hasn't it? Tonight, what do we have in store? Well, I'm going to talk a little bit about my travels. I had a couple of interesting European travels, and I'll talk about two of them, which involved science in Sweden and Finland. I have a lecture, mini lecture queued up if you want. We'll see if there are questions. If there are not so many people here, there are not going to be so many questions. So we'll just do a mini lecture. But don't forget to hit the like button right away so that we can attract more people and tell them all about good science. Because as you know, even the New York Times doesn't know what correct science is, apparently. We're going to talk about that a little bit tonight. This horrible, horrible opinion piece in the New York Times a couple of weeks ago. I'll talk about our response to that as well. So lots to do. And of course, don't forget your questions. I always like hearing from you as well. Here I'm in New Jersey tonight. It is 30 Celsius. The U.S. is the only frickin' country in the world that does not use Celsius for temperature. I saw a post on Instagram. Countries that use Fahrenheit, little colored U.S. That's it. And being in Europe, of course, everybody uses Celsius. So I felt right at home. There's this complaint that it's too compressed. Well, the Europeans don't seem to care. The rest of the world don't seem to care. My God, folks, get with it. This is cowboy American vision. Sometimes it serves you wrong. Okay, let's see who's here. So as I said, our moderators, we told you where they are from. Lise is from Columbus, Ohio. And I have to tell you that being in Sweden and Finland, it was like 25 C most of the time. And you figure out what that is. You don't have to convert it. You just have to build into your head a sensation of what 20, 21 through 30 feels. You don't have to convert. Oh, it's going to be 25 today. I know what to wear. Conversion is not the right thing to do. Red Hen 689. I normally listen to from PA, but I'm in London now. All right. It's wonderful to travel. Betty is in Washington Heights, New York City, not too far from where I spent many, many years working at Columbia University, Washington Heights. Christella is in Ohio. Kang is in Chicago, where it's 30 C and partly cloudy. Mark Martin's in Tacoma, Washington. Hello, Mark Martin. How are you? Frank is in Santa Barbara. Patty is on the New Hampshire seacoast. Patricia's in D.C. Brian, who I met last, I think, November-ish, December-ish, is in Montreal, Canada. Abdulaziz is from Saudi Arabia, where I'm sure it's a lot hotter than it is here. Oh, boy. Jonathan's in is in Encinitas, California, 24C. There you go. And Jonathan knows to use 24C. Out walking. Oh, look at this. Dean is from Lithuania. Looks like a nice cup of cappuccino. Welcome. Abdur Rahman is from Egypt's north coast. Rima is from Iowa. Kathleen is from the Jersey Shore. El Cio is from Belém, Amazon, Brazil. Cool. Hot and raining. Claire's from the UK. John is in Minneapolis. Cheryl is in Santa Rosa, California. Artemis, also I met last year in Montreal. Mike and Lori in Seattle, Washington. Laura is in Connecticut. One thunderstorm just passed. Yeah, apparently it's due here as well. And thank you very much. Yes, I missed being here. I have done most of the podcasts, as you saw. I was able to do many of them on the road. And yeah, there's a skill to doing podcasts on the road. First of all, the first requirement is desire. And there's no question that I want to do them every week. And so it's just a matter of bringing enough equipment, a nice microphone with an interface for your laptop. I used the camera on my laptop and I brought a light. So I have this little light, which I'm using tonight, actually. I'll show it to you. It's pretty cool. Okay. It's a nice little light and it's giving me some fill tonight because if I don't have this, I get the Rembrandt look, right? I don't think we need the Rembrandt look tonight. And you can dial up the intensity. You can change the color from, warm to cool. And it's really light. It runs off USB-C power. And that was all I needed. Now, of course, I did record a couple of podcasts in Sweden and Finland, so I had to take other equipment with me. And that I used a bit as well. Okay, let's see. What else do we have? Did Liberia switch to Celsius? On the Instagram map, I should have been able to see Liberia, but I swear that all of the world was Celsius except the US. EHS Talent is in Pennsylvania. John Sheehan is in Port Townsend, Washington. If Amy Rosenfeld is in here, it was good to see you recently. I don't know if she's here, but yes. You, of course, saw Amy sitting in for me on Clinical Update with Daniel Griffin. That was a lot of fun. And MN is from near DC. Artemis, having grown up in Europe, C is the only temperature unit that makes sense to me. Hat is in LA where it's 25C. Cairo in New York City says I can do conversion in my head in 3 seconds. I'm completely fluent in metric and convert meters to yards, something to miles instantaneously. Yet when I'm in when I'm, I guess it's later. Okay, Mr. Doreen is here from Elgin, Illinois. Richard is in El Segunda, California. Mark says Encinitas is nice. I lived there for several years. Fish tacos. Okay. Fish tacos. Who else do we have here? Pete is back in London. From Dorset, Silent Rudders in Kansas. National Geographic states US, Burma, and Liberia use the Fahrenheit scale. Still pretty overwhelming, Frank, right? Most of the world using Celsius. I don't know. Grace Rosella is in Melbourne, Australia. Is my volume too loud? I can lower it if you'd like. According to my meter here, it's perfect. It's Goldilocks, not too hot, not too cool, but let me know. Hat is a twiv aholic that like a latent virus is reactivated with you. Great to have you back. It was great to see Amy on Clinical Update. All right. There we have it. I did see a number of other questions here. Karen's playlist is from northern Maine. Okay, the volume seems to be okay. I'm always happy to fix things. You just need to let me know. Audio seems fine. Rima is going to use Kelvin for temperature. Good luck with that. Nobody will know what you're talking about, but I guess it doesn't matter, right? Well, you just need to look at your phone. I have Celsius on my phone. People ask me what's the temperature. I tell them in Celsius. 29C currently. Figure it out. You know, the U.S. tried to do the conversion years ago, but it didn't work out. People are recalcitrant with science. Here in the U.S., we have a science problem. A couple of questions here I'm going to answer before I jump into some other things. And you guys wanted to know what this photo is right here. I'm going to talk about that. Someone had a guess. It is a lock, right? The kind that boats go in and either go up or down, but I'll tell you where it is. It's something I've wanted to do for many years. I'll tell you all about it. Kevin said, I saw a paper that said a virus was discovered in the Mariana Trench. It infected bacteria living there and actually made them more fit to survive down in those extreme conditions. And that's very interesting. I'm not surprised. There are viruses everywhere. Wherever there's life, there's going to be viruses that infect those living things. So it's not surprising. But in this case, you see it's beneficial, which is very cool. And I think we're going to see more and more of that, viruses being beneficial to their hosts, because in most cases they probably are. They coexist very well. Mark sent me some virus toys to throw at anti-science people. I should have brought some down here. They're squishy, soft things that look like viruses for sure, in all different colors. They're very nice, Mark. Thank you very much. There was another question here. Let's see. Kang wants to know, are there any studies on SARS-CoV-2 binding to ACE2 receptors affecting the levels of angiotensin and causing vascular disorders? You know, I bet there are. I bet there are. I'm looking it up. But I'm not aware of the studies. So does ACE2 cause vasoconstriction? No, that's a different question. You know, I have nothing has jumped out at me that has been impressive. There may be some minor studies that aren't really impressive. In other words, nobody's followed them up. Who knows what they mean? There are obviously vascular disorders in COVID, but they seem to be cytokine based, not caused by ACE by spike binding to ACE2. That's an interesting question. Mark says, I hear that people who work on giant viruses really don't like the term gyrus. Yeah, many years ago, someone came up with that giant virus, right? Gyrus. Well, it's meaningless, right? It has no taxonomic significance whatsoever. What could I liken it to? A car. Doesn't tell you anything. Or maybe even SUV. I don't know. No, I don't like the term. It's useless. Giant virus discovery was really interesting because they were bigger than anything we'd seen before. But, and you know, they classify based on nucleic acid sequence homology together. But as you can imagine, there's also an overlap. There are big ones, really big ones, and not so big ones. So classifying just on size doesn't make any sense to me. But what do I know? I'm just a virologist. Abdur says, I believe the attacks on medical authority is part and parcel of the attempts to discredit any body of authority to allow hucksters to run scams. I always wonder what the motivation is for people to promulgate false information. So, as you know, a few weeks ago, Alina Chen published a opinion piece in the New York Times saying 5 reasons why SARS-CoV-2 likely came from a laboratory. And it was completely wrong. Every one of her 5 points were wrong. They provided zero evidence that the virus came from a laboratory. And she just ignored all of the evidence that the virus came from nature. Saying, for example, it's never been isolated from animals in the market. Well, guess what? They killed all the animals on January 1st, so there was no opportunity to isolate virus from them. Whereas for SARS-1, the outbreak continued for 7 months before they closed the market down. So there's ample opportunity to sample the market. But we have other evidence that the market was involved, which she ignores. She says there's no good evidence that the market was involved. This is 100% BS. There are a couple of papers that show that the market, the Huanan market in Wuhan was the early epicenter, that both lineages of SARS-CoV-2 were circulating there, that environmental samples are positive not only for SARS-CoV-2, both lineages, but also mammals in the southwest corner of the market that are known to be infectable by SARS-CoV-2. And she just throws that up saying there's no good evidence. Why? Because it's devastating to her hypothesis. There's no way to get around it, folks. You can say all you want that the data are no good, that there are problems, blah, 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 but you'd be wrong in every case. The data are devastating and paint a very clear picture of where this virus came from. So this is a waste of everyone's time. I agree that maybe on day one, we could say, well, let's make sure where this came from and look. But in the intervening years, data after data has come out supporting a natural origin. Zero data. And people write to me and say, but you're ignoring this and that and the other. I'm not ignoring anything. There are no data. It's just innuendo and hunches and suppositions and accusations. That's not data. Oh, the Wuhan Institute of Virology was working on coronaviruses. Okay, nobody's hiding that. But you know what? They weren't working on SARS-CoV-2. Do you understand that? SARS-1 is very different. And that's what they were working on. So this just is crazy. I saw this article when I was in Finland, and we were preparing for the next twiv. It was early in the week. So we have to address this. And so when I got to Helsinki on Friday, I had a free day on Friday. I spent most of the day writing a rebuttal that I would give a coherent reading of during twiv. And that twiv we recorded at 3 p.m. Eastern. It was 10 p.m. Helsinki time. And then I decided to split it off into its own part also, a 55-minute just evidence that SARS-CoV-2 came from nature. You know, people write to me and say, you're wrong. I'm not anything. All I'm doing is showing you the data, okay? It's not about being wrong or right. Look at what the data tell you. Oh, but I don't like that data. It wasn't done right. Well, it's too bad if you don't like it. It was accepted. No one's applied to retract the papers. You're wasting everyone's time and making political antagonism, and we're never going to know for sure where the animals were that were positive because China is now resistant to all investigations because the U.S. has politicized it. Okay? Good job, folks. Really smart. I don't know what you're thinking about, maybe just your pocketbook, but it burns me up. All I care about is the science, folks. If the science said it came from a lab, I'd tell you that, but there isn't anything. And I don't understand what people are thinking when they tell me, Vincent, you're wrong. You're 100% wrong. It's not me. It's the data. Anyway, I don't know what you folks feel. If you think it's a done deal or you're sad that the New York Times published that. I'm really sad that they published that crap. You can publish an opinion piece. That's fine. But there's no basis for her opinions. And what she's saying is that this happened, but she says the vast majority of evidence indicates it came from them. No, that's not even right to say that. The vast majority of evidence says it came from nature. Why should you be able to lie in an opinion piece? I just don't understand that. Respect for New York Times, 1000% dropped. I canceled my subscription last year. You know, I do support journalism. I think it needs to live. I thought the coverage of COVID was miserable, and I think this is horrible. I don't see why they did that. So that's how I feel about this, folks. And maybe you have opinions as well. Okay, let's see. Let's go back to your questions. Oh no, let me talk about my trip a little. Okay, then we'll come back to your questions. Okay, so the ultimate goal was a meeting in Finland in a small town 3 hours north of Helsinki called Jyväskylä. And okay, I had a power outage because it's thunderstorming. Everybody hear me again? I'm seeing myself on the stream. Okay. Yeah, it was an electrical. I don't have a backup power. I do have surge suppressor, but obviously it just kicked out and shut down the computer. Okay, what was I saying? Let's talk about my trip. So I stopped in Sweden first. Let me show you some pictures. I have a couple of friends in Sweden, and one of them is Erling Norby. Erling was a virologist for many years. He was on twiv a couple of times, and he's interesting because he served on the Virology Nobel Selection Committee for many years. So he has a lot of insight, and he writes a series of books on the Nobel prizes. You know, every 50 years after the prize is awarded, the archives of the Nobel decisions are opened up. So he looks at them, and of course, he has to translate them from Swedish, and he writes books about them. And so I met him. I came in and met him and my friend Agnes, who I'll show you in a minute, and he brought us to the Royal Swedish Academy of Sciences. That's this right here, this building, which is, as the name says, a institute that looks after science and history of science in the U.S. and plays some role in the Nobel Prizes. So this is the opening for that. And then Erling, well, here's Erling and a portrait of him. Since he was I think he was the secretary to the committee for many years, he has a portrait of himself in this in this Royal Swedish Academy of Sciences. Now, this Royal Swedish Academy of Sciences has some role in the selection of Nobel Prizes, and so that happens actually in this room. This is, I believe, they have deliberations here around this table, and I believe some of the prizes are announced to the press here, at least initially, and then they're further discussed somewhere else. It's kind of a neat room. It's got a lot of history of a lot of old people who on the wall there who chaired this institute for many years. Anyway, so there's Erling. He has an office in the Swedish Academy of Sciences. and it's where he writes his books. It's very, very cozy little office. Okay, now this is Agnes, who I met many years ago in Europe at a meeting. She's an MD working at the Karolinska. She's a clinical microbiologist, and it's her husband there. And I think I met her in Rotterdam in 2019 or 2018. She said, "If you ever come to Sweden, let me know." So I went to Sweden in 2019, and I informed her, and she brought me all over the place, me and Erling. And then this time I said, "I'm going to Finland. Should I stop by?" She said, "Absolutely." So she arranged a series of podcasts with scientists at the Karolinska, which will be published over the coming weeks. But if you've ever been to Stockholm, it's a lovely city. This is where the sun is starting to set here. There's a lovely river running through the town. It's a very cosmopolitan city. It's very accessible and very easy to get around. I like it very much. I'd go back easily. Now, when I was there in Stockholm, I gave a talk to infectious disease doctors. Here is my audience, and they had all said, "Hey, I heard Racaniello's coming to Stockholm. Why doesn't he give a talk?" So I gave a talk about polio, and they were all very, very interested in that. So I think that was cool. Then I did a podcast with two individuals that'll be coming out shortly. One of them all about HPV eradication in Sweden. You know, they don't do pap smears anymore in Sweden. They just do swabs and they look for human papillomavirus by PCR because that's the only thing that matters for cancer. Doesn't matter if you have morphological changes in cervical cells due to other things because they don't cause cancer. The only thing that causes cancer is HPV, and so you look for that. It's a very interesting approach. No more pap smears. Can you imagine? Anyway, I recorded that podcast in a room called the Ralph Steinman Room. Every room in this building is named after a different scientist, and I thought that was cool. Ralph Steinman, of course, an immunologist at the Rockefeller University for many years, and was awarded the Nobel Prize for discovering dendritic cells. There are two campuses of the Karolinska Institute. There's one in the city. It's called Solna, and that's this building here, and the one where the Ralph Steinman room is in Huddinge. That's just a little bit outside the city. And this is a building called the Biocentrum, which is all labs and has an internal channel, as you see here. It's really lovely. It's made so well. Everything is transparent. The noise is muted. And on the bottom floor, they have the Cafe Biomedicum, where people sit and eat and chat. I think it's really, really, really well done to foster interactions. I did a podcast with Leah. I've forgotten her last name. Let me look it up. Leah Bruckman. I believe that's her name. Let's look. Let's search for this. This is not nice not to know people's last names, right? Judith Bruckfeld. Not even close, Vincent. Judith Bruckfeld is an MD who works on long COVID. All right. So that's going to be interesting to you. And then I did also a podcast with Niklas Bjorkstrom and Joachim Dillner. And all of those will be coming out in subsequent weeks. So while I was in Stockholm, I did a lecture and two podcasts. All right. So then that last Saturday, Agnes took me on a boat trip in a canal. And there's a canal that runs through central Sweden. It's been there for many years. And you can see there are multiple locks to get the boats up and back down again. And when I had visited years ago, I had seen this and I said, I want to go on one of those boats. So she said, okay. So we took a full day trip on the canal. And that's where this picture comes from. So this is quite a large boat. I would say, I don't know, 60, 70 feet. It was motorized, of course. It was a full day excursion. And, you know, the boat goes, this one is a lock where the boat goes in and then the water fills the lock. The boat floats up and then the locks open, the gates open, and the boat moves to the next one. And so this is, as you can see from, let's see, this map, these are the names of the small towns along the way, if you want to know where this is roughly in Sweden. It's about 2.5 hours out of Stockholm. And it's actually a part of a system that can go from one side to the other, one body of water to the other. So it's pretty cool. Anyway, that was a fun day. And if you're wondering, this CAP, ASCAP is an association for music, and I don't know what it stands for. ASCAP. Let's look it up. American Society of Composers, Authors, and Publishers, right? It's an organization that licenses performance rights. So somebody made a cap and it's perfect, right? It's an ass cap. So I'm an ass and I'm wearing a cap. You get it? Okay. All right. That is Sweden. And then onward to Jyväskylä, which is 3.5 hours north of Helsinki. So a flight from Stockholm to Helsinki and then a train from Helsinki north. And this was the reception on the first day of the meeting. You notice Dr. Amy Rosenfeld here. She attended this meeting as well. And these are what picornavirologists look like. These are people who work on picornaviruses like poliovirus and enterovirus. It's a very beautiful venue. Here's that right outside the window is this lovely lake, and I have a picture of it here. Beautiful, very beautiful country, gentle rolling hills. Finland's a big landmass, but there are only 5.5 million people. It's amazing. New York City is 12 million, 11 million, right? So that was fun. And let me tell you about the meeting. So I had a badge, of course, which has my affiliation, but I like that they also put This Week in Virology. And by the way, the name of the meeting is EUROPIC, European Picornavirus Meeting. And they have one every couple of years. I've been to many of them. Here's one of the scientists I knew at the meeting, Elisabetta. She's an Italian structural virologist working in the UK who I've known for a number of years. And here's a poster that comes out of Amy's lab and Mike Strauss, her structural collaborator. It's structural work. And you can see my name is on there because some of this was done in my lab. And maybe this is the last poster I'll ever have. So that's pretty cool. Dinner, lunch, breakfast, all happening at this very nice conference venue, as you can see. Lots of opportunity to network and talk to people. I did an episode of twiv at this meeting with Caroline Taparel and Tapani Hovi. This is already released, so you can watch that where they talk about their work. And then one thing that was very interesting during these meetings, the organizers like to have the people do things so that they learn something about the country they're in. And one afternoon, they took us to a museum that showcased the work of Alvar Aalto, one of the masters of modern architecture. I'd never heard of this guy before, but if you know his furniture, you know him totally. And he invented the kidney-shaped swimming pool. And in fact, as you know, here's the museum. They were giving our group a little bit of a lecture here. It's a lovely museum. And this guy was talking all about the history of the museum. I was up on the upper floor, which has kind of an exhibit of skateboarding. So why skateboarding? So as I said, he invented the kidney-shaped swimming pool. And of course, skateboarders immediately drained them and skateboarded in them, had unique shapes. So part of this museum is devoted to skateboarding and various skateboarders. And there's this wonderful picture of this guy, doing a maneuver on his skateboard that I had to have a picture next to. It's not clear who's upside down here, right? But I really like this picture. One thing that's interesting about Finland is that all the trains have my initials on them, VR. So you can't beat a country like that. Not all the trains, but this is a particular train line. Both freight, as you can see here, and passenger train line. And then on to Helsinki, just for 2 nights, and I ran into this interesting, store here, the Stupido store, so I thought I should have a picture in front of it. And, also Helsinki is a very cosmopolitan town as well. I'm having dinner on the sidewalk here. It was very good food. It's very accessible. Nice to walk around. The weather is very mild at this time of year. Of course, it gets very cold, but in the winter, the interesting thing about both Helsinki, both Finland and Sweden, it never really gets dark. So the sun sets about 11 p.m. The skies start to dim, but they never get dark. And then at about 1:30, 2 a.m., the sun rises again, and that's it. So you never get a dark night. Okay, so that was my Scandinavian journey. I really like it very much. I hope you enjoyed the little slideshow. Now you know where that photo came from. So it is a lock, yeah. Mark says, "How many sets of glasses do you have?" I have a lot. They're mostly these cheap things that I buy. This is like a $2 reading glasses from Amazon. Like I got kind of a clearish blue here, and here's the original blue that I started with. I have a bunch at the incubator as well. Here's a red pair. I have these kind of funky ones that I don't really wear, but they're kind, really big circular guys. And then these are my regular glasses, but I can't wear these. I wouldn't be able to see the screen now. But I would say I have at least 20 different pair, but they're very cheap. Okay. You can buy them for $2 each at Amazon. Okay, let's go back to our comments here. Oh yes, Twivaholic. Oh, we have a new addition from Brazil. Klaus, welcome to the stream. Abdul, thank you for your contribution to science communication. By the way, if you like our work, both here on these streams and also on our podcast, we'd love your support. You can go to microbe.tv/contribute. Oops, wrong finger. See? I don't know why Vincent here is. We don't need Vincent email. And then you can do it by Venmo if you want. Or here in the live stream, there's a chat, super chat, where you can push a button and dial in some numbers, which is what Abdul Aziz has done. So thank you very much. Really, really appreciate it. A friend in the UK told me that her aunt said they should wait until all old people have died before going metric. Mark Martin is trying to get more virologists on Matters Microbial. What's the problem, Mark? Do they not answer you? Do you need some assistance? Plenty of good virologists out there. Why don't you just drop my name? Maybe it would help. What do you say that over 92 animals tested positive for SARS-1, but over 400 from SARS-2 and 0 positive? Patricia, they removed all the animals on January 1st. No animals were tested that were in the market. As I said before, SARS-1 went on for 7 months before they closed the market. So there's ample opportunity to test many, many animals, and they found positive animals in the market. Now, the Chinese government subsequently went out into the countryside and tested animals, and they were negative, but those were not animals that were in the market. So what's the point of that? And the number of those animals that would be infected would be vanishingly low. So what you're writing here is tremendously unfair because no animals were actually tested in the market. And so this is not a point that can be used to make any reasonable argument for the origin. Any plans to experiment with shorter form content? The discussion and the university lecture formats are great, but there might be scenarios where more highly produced content would reach other audiences. Absolutely. We're only a couple of years old, and we're doing fundraising, right, to try and get enough money to hire people to help us. And as soon as we get to a certain amount, we'll start bringing people on board who can do short form. But we have done some 1 minute type videos for TikTok and Instagram and YouTube and so forth. But yeah, I would like to have like 5 minute videos. I've explored myself in making very short videos where I explain something very quickly. I agree, you can reach different people. The long form got us into this business, and for that I thank them. But I think short form will reach more people. For example, so I have I have a social media individual who works with us, Kim. I said to her today, I want to make a 1 minute video explaining why SARS-CoV-2 came from nature, not from a lab. Do you think that's possible? She said, absolutely. You can do it. Right? 1 minute, all the people have to do is listen for a minute and they will be convinced that it didn't come from a laboratory. So I think that kind of thing is possible. So yes, we're going to do that. In the UK, they established the British Metrication Board. They printed billions of leaflets and convinced all Brits to go metric. Yeah, well, the desire wasn't present here in the US. I remember when that happened. I was quite young, and I was very excited about going metric, but sadly it didn't work. Yes, Mark, today I recorded a Beyond the Noise with Paul Offit. He wrote a column on Monday, debunking the opinion piece in the Times, and he wanted to do a video on it, so we did that today. So the first question I asked him was, what was your reaction when you saw that opinion piece in the New York Times? And he said, I was angry. And he explained why. That'll be out next week, so you can check that out. A dengue fever epidemic is going on in Brazil, at least in São Paulo. Many people getting ill even during a drought. Enter the entomologists. Yes, dengue fever spread by mosquitoes, of course. Even your first dengue infection can be very unpleasant. Freedom of deception should not be allowed in the Constitution. It's natural for humans to deceive one another, right? Scientists are human beings. The good part about science is that we can check things. When people respond with conspiracies, I get irritable. Yeah, so one of the here's the idea of the lab leakers, okay? So early in 2020, like January, when the first sequence of SARS-CoV-2 genome comes out, Eddie Holmes, Christian Anderson, Bob Gary, Marion Koopmans, Ron Fouché, they all start to have Slack conversations, which have subsequently been released through a Freedom of Information Act. You can go see them talking and worrying about where this virus came from. And they're saying, oh, I'm worried about that furin cleavage site. It's not present in any other SARS-like coronavirus. And then this thinking evolves, and they publish a paper, a subset of them publish a paper called Proximal Origins, which says it most likely came from nature. This was very early 2020. So the lab leakers say they had a conspiracy. They knew initially it came from a lab, and then they decided to cover it up and hush, hush. And only when they got the Slack conversation does the public realize that they're colluding to cover this up. And Tony Fauci is involved as well. This is the biggest pile of horse shit I've ever heard in my life. Why the hell would you do that? Why would you cover up for another country? None of the scientists were even from China. Scientists want to know the truth. This is just BS. And anyway, you can't even get scientists to agree on things. You think they're going to agree on cover up? Oh my God, this is so stupid. They changed their mind because the data supported a natural origin, not a lab leak. There's no collusion. no covering up anything. But that's the thing, as Mark says, people say about conspiracies because they don't understand. So they have to have an explanation for where something came from. Oh my gosh. Crazy stuff. Back to metric. My only beef with metric is that 10-degree increments in F are incredibly useful for everyday life. Yeah, but most of the world doesn't have an issue, right? They deal with it. I agree with what you're saying, but most of the world deals with it. So, I don't know what to say. Abdur says it's an authority thing. It's inherently political in a way. People challenge anyone's authority to assert themselves in the speech market so they can peddle nonsense since they have no credentials. And of course, the Republicans jump on this, and now they want to regulate science left and right because obviously this virus was created in a lab, and we can't let these scientists get away with that anymore. This is just the ultimate BS. The Republicans have no freaking idea what they're talking about when it comes to science. With all the inquiries they've done, they haven't added anything. There were 3 intelligence community investigations into the origins. All 3 of them said the lab didn't have a virus close enough to SARS-CoV-2 to be able to make it. All 3 of them concluded that. So why are we ignoring that? Jesus, unbelievable. Once upon a time, the New York Times did good journalism. Now they still occasionally publish a good piece, but it's very haphazard. I might agree with that, but I don't read it anymore because I'm really disappointed. And I don't want to get my science from the New York Times anyway. Conspiracy theories are sexier than science to some. Outrage is addictive. Okie dokie. So here we go about the metric system. U.S. almost switched to the metric system in 1902, but the legislation was defeated by 1 vote. Congress passed the Metric Conversion Act in 1975. Besides the critical papers with respect to the Wuhan market origin, there's a vast supporting literature on coronavirus variation and spillover. Yeah, every human virus has come from an animal. Every single one. No exception. Because the variation you need to do that, to go from animals to humans, can only happen in animals. It's just crazy to think you could engineer. And by the way, these lab leakers, they're not even sure they're not sure if it was engineered or maybe it just was a sample that got out. They don't even know because there's no evidence for any of it. Just hypotheses without any data. Okay. GSD, Jonathan on twiv 1125, you brought up ideally you would want to vaccinate pigs to mitigate risk from swine flu. Cow vaccination came up too. What if costs could be managed? So Angela pointed out that vaccinating cows and pigs would be very expensive. And I was thinking about this further. You have to test the vaccines to make sure they work, right? You can't just make it and give it to pigs and assume it's going to work. So how do you do that, right? Let's say that the H1N2 pig virus that we talked about. You'd have to do challenge studies with pigs. Yeah, it's going to be very costly. And the same with cows. You're going to have a set of cows and infect them, establish infection in the mammary glands, and then have another cohort that's vaccinated. And this is not going to be quick. And who's going to pay for it? Right. So you say, what if costs could be managed? Well, yeah, if you could manage, I have no idea. I'm not a cost person. I have no idea about costs. But yeah, I suppose if you could manage it, but it just seems like quite an undertaking to do that, which is not to say that we don't immunize certain animals, right? Fish are routinely immunized against fish viruses. 1 fish at a time. You pick it up, you put the syringe in it, inject, and put it in the bin. So it's done. Why can't Denvaxia be given to people who have never had dengue? Because the type 2 component is not good. There are 4 serotypes, and the type 2 serotype in Dengvaxia does not give you a good immune response. So you're basically like a dengue naive person, and that causes severe type 2 dengue. Former CDC guy Robert Redfield also keeps saying lab origin. This is unbelievable. He's an idiot. You know why? This is the guy who said HIV was a gay disease back in the 80s because he was involved with HIV. So you should not listen to a word of what he says because obviously that was wrong and biased. And so he just doesn't know. It's a political issue. It's totally political. I guess all of you are converted. I imagine you met a lot of interesting researchers during this trip. Can you tell us who stood out, new friend or old, what their current research is, why it caught their interest? Well, there were some. So, like all fields in science, there's turnover. So the coronavirus field, I've been to 10 of these meetings every couple of years. So I have seen a whole generation of coronavirologists move on, including myself. I don't have a lab anymore. I went to do a podcast, which I think is a good reason to go for sure, but I'm not a working scientist stiff anymore. But all the people I knew, Jeff Almond, Phil Miner, Eckhard Wimmer, Dave Rollins, many more not there. The only senior person or couple actually did a poll at the beginning of TWIV who's been more than 10 Europics, or just a handful of people. One of them is Raul Andino from UCSF, who I know very well. He was there. And also Frank van Koepenveld from the Netherlands. So very few of the old guard, a lot of younger people, which is good, who I don't know. But that's the way a field should be. And I can, like Elisabetta, she's I would say a mid-career scientist. I know her. I can talk to her. I can talk to anybody. I can go up to anyone. Just talk to anyone. And they all get communication. They all seem to understand TWIV. And so that's a nice bridge that I have with everyone. Okay, this is just where I had a hiccup. Jeff writes, RE Lab Leak. The problem is the GOP and their successful long-running attacks on government to make it appear that it can't do anything right, in this case, supporting Chinese research. Yeah, it's just an opportunity for them to further attack the government. And as you see, we discussed on TWIV, now there's a proposal to revamp the NIH as if it's been doing wrong things. And what they point out in this dumb report, we can't have labs doing gain of function and potentially making pandemic viruses. This is just idiocy. I was brought up in an era where you used your brain to think about things logically, and we don't do that anymore, especially the politicians. All they want to do is advance themselves, and they'll say anything that comes to their mind, whether it makes sense or not. And when it comes to science, it makes no sense. They have no idea what they're talking about. Please shortly explain how the hospital lab machine counts hepatitis B virus viral load into millions per mil. Is it using light or electricity to count the virus pieces? What it's doing is it takes a small amount of blood, extracts the DNA, and does PCR, polymerase chain reaction, to amplify the PCR. And then it can express the number of DNA, viral DNA copies, in terms of copies per milliliter, right? And so you can have very low, intermediate, or high copy number, but it's a PCR-based mechanism, a DNA amplification. Lise sent an email to New York Times about a counter opinion. I would like to write a counter opinion, I will write a counter opinion with a coronavirologist co-author. I have a lot of notes from the twiv episode, so I don't think it should be take too long. But you have to write things very nicely and clearly and succinctly, and so it could take some time. But um, I have published one opinion piece with Amy Rosenfeld a couple of years ago, so we have an in there. So we're going to try that. Yes, so we're gonna try and do that. The GOP created an environment in which people remark how government can't do anything right, only private sector can. Yeah, they want everything privatized. The postmaster is running the post office into the ground, the postal system into the ground, because he wants it all privatized. It's pathetic. I love the post office. People there work hard. They do a good job. It's just screwy what he wants to do. No, no, no. I mean, you can't even hide it. It's quite clear that he's trashing it all left and right, and then it'll be privatized. Hans writes, since a virus is either pathogenic or not, and since virulence is a measure of said pathogenicity, shouldn't it be highly virulent, H5N1, instead of high pathogenicity, H5N1? Yes, so virulence is the quantification of pathogenicity. You're right. It should be. It should be HVAI, high virulence avian influenza. But often scientists don't abide by conventions, right? I mean, on a daily basis, I see this. So, for example, today on Twivo, they're talking about amino acid mutations, right? Because it's not right. A mutation is a change in nucleic acid. An amino acid is an amino acid change, not a mutation. But scientists will argue with me. Oh, don't be pedantic. Doesn't matter. Yeah, it does matter. Your nomenclature matters because you all have to be saying the same thing. And so, yeah, you're right, Hans. Good point. Good pickup. And Jeff continues, the GOP has also successfully cut funding of many social programs to the point that they don't function well, thus adding fuel to the government can't do anything right. As an example, the Postal Service being run into the ground. Yep. There's a saying in the newspaper publishing, if it bleeds, it leads. Same can be said for rage bait articles and op-eds. How pathetic. Just pathetic. Paul Offit said when that was published, that many of his friends called him and said, "You see, I told you it came from a lab." It doesn't prove anything, folks. Oh my gosh. End Root says, okay, so a lot of times, and this is in response to another listener, a lot of times when people say viral load, they're assuming the number of viruses is correlated with the amount of nucleic acid detected via PCR using fluorescent compounds, right? It's not always. That's the greatest example is during the COVID pandemic, the variant arises and you get more PCR product, and therefore it must be more infectious virus, and therefore it must be more infectious and transmissible. Logic flaws left and right. No. In fact, different viruses, alpha, delta, gamma, omicron, make different amounts of RNA. And it doesn't correlate with the amount of infectious virus. Did you think about that? No, because you're not a virologist. So you cannot assume, unless you do a calibration curve for your specific virus, not a variant, to make sure that there is a correlation between the two. You don't know. Artemis says, I read an article saying that screening for HPV should be followed by a pap test in case PCR comes out positive. The rationale is that having HPV does not always mean you will get cancer. So what my guest told me, no, they don't do this in Sweden. No more pap smears. If you have a positive HPV PCR, they will come in and look at your cervix for a lesion. If there's a lesion, it will be treated, by laser ablation or whatever. If there's no lesion, they send you home because there's nothing to do. And then you have a swab periodically and you see what happens. He said many cases are cleared. Infection clears after a certain point, so you don't have to worry about it. So there's no need to do a pap smear. So I don't know why they would do this. Because if you're PCR positive, you need to go in and look if there's a lesion. And he said to me many times, excuse me, there is no lesion. And so there's nothing to do. There's no lesion. There's no lesion to progress to cancer. End of story. So when you take a pap smear, right, you're looking for abnormal cellular architecture, right? And there are different things that can cause that. One is HPV, and that's the only thing that can lead to cancer. And then there are various protozoans, for example. Doesn't matter. They don't matter. You're not going to get cancer from them. So you don't need a pap smear. The only thing you need is a swab. And the whole point was during COVID, people couldn't come to offices to get pap smears, to get swabs for HPV. So they started mailing self-swab kits to homes, and they said it worked brilliantly. People mail it in, they do a PCR test, and the people who are HPV positive, they can follow up on. Thank you, John, for your contribution to science communication. Really appreciate it. So Les tells us that the Finnish state railways, that's VR, Suomen Valtion Rautatiet VR. Okay. Gabriella asks, I wonder which relevance could it have an Epstein-Barr test that resulted in exposed but not active? If almost everyone has been exposed, what do you think? So I think what you're talking about is probably a serological test, right? Which means you have antibodies, but then you don't have any genomes. So the virus isn't replicated, but you have antibodies to EBV, which is the case. Most people do, right? But not everyone is replicating virus at any given time. So that's the difference there. Yeah. Pull off, and now we're talking about length of podcasts. Pull office videos are short because I make them short. I make up 10 questions and I keep it to less than 15 minutes. I make an active effort to focus on the most important questions. He doesn't give very long answers, which is great. I follow up sometimes, but it's because I deal with it. And someone else might let it go for an hour. I don't know. But I thought from the beginning it should be 15 minutes. And mostly it is, yeah. Is there a reason why the HPV vaccine is given up to a certain age? Yes. I asked my podcast host this very question. I said, my listeners all want to know this. It's like, why is it up to 45 years of age? He said, simple. It hasn't been tested in older people, so you can't give it to them. That's it. Simple story. John wants to know how much impact has the U.S. government had on EcoHealth Alliance and surveillance of emerging infectious diseases? Well, they took away their bloody NIH grant, right? So they can't do that work anymore. And in fact, when they restored it to them, which was just for a number of months, they said you can't work with the Wuhan Institute of Virology. So that's done, at least in China. Now, they had programs elsewhere for sure, and presumably they're continuing. There are other research units that are doing this as well in many different countries. It's not huge because it's hard, surveillance is hard work, and people don't want to do it. But obviously, it's essential, and there's a paucity of it because the papers on surveillance are very few and far between. We try and do them as much as we can, but there aren't that many. Are there any plans for a this weekend mycology fungi? People have asked for it for years, but no plans. The way I figure is that we can do fungal papers in this week in microbiology, right? Yeah. In fact, at ASM in Atlanta last week, TWIM interviewed Arturo Casadeval all about Candida auris. And Michael Schmidt and Mark Martin were co-hosts for that. So look for that in a couple of weeks. What's the average length of a TikTok video? Video sound quality there doesn't need to be of super high quality. Well, it's interesting. So they're all I'd say they're less than a minute. You can make longer ones, but if you look at the most popular ones, they're less than a minute. The video quality is actually quite good because cell phones take great video. Not the audio less so, but doesn't seem to matter. I don't know what is the key. I see people on the train, they're all looking at TikTok. They're just flipping from one to the other. They see it for a second and they flip up. And then every now and then they stop and watch one. I want to know what they're watching. They're not watching science, I bet. But what is it? What is it that attracts someone to stop and watch a TikTok video? Is it something crazy? Is it something funny? Probably. People want to be entertained on the train, right? It's a fundamental aversion to being educated because it's hard work to use your brain. I understand that. But folks, it's so important so that you can figure out your lives. Please do go into the short form, but don't minimize long. It allows for fleshing of views. More intricate arguments lets people explore their arguments, counter arguments, and points of view. Oh, totally. I would never give up the long form. I could see one day microbe TV is all these young, excited people and one old guy, me, and I'll be the only one making long videos. It's okay. I will stick to long videos because I think they're great that you can have a deep dive, right? 55 minutes with why SARS-CoV-2 came from nature. Where else are you going to get that? Yes, so we will for sure keep the long form, but we'll just expand. John asks, isn't the second bout of dengue usually fatal? It's more fatal than the first, but not always. And that's not 100%. It can be accompanied by hemorrhagic fever and shock syndrome, capillaries breaking and bleeding and so forth. So it has a higher fatality rate, but it's not 100%. Why did at least one that we know of NIH scientists brag about being able to avoid FOIA? All right, so the only person I can think of is Dr. Fauci's assistant who was using his personal email address to avoid FOIA, right? I don't know. I don't know why he did that. It's the only one I know of. Please do a show or part of a show discussing the lack of evidence that the mRNA vaccines increase risk for cancer. Dr. Campbell, who has for months pounded on this issue. So Campbell doesn't know anything about mRNA vaccines and cancer. He's pushing fake hypotheses that aren't supported by any data. And yeah, I saw a video of his pop up, right? mRNA vaccines, no. And I thought I should probably counter it. But that would require that I watch his drivel. And I just despise wasting time watching his drivel. Is that right? Yeah, it's a waste of my time, but I understand I need to do it. Should news outlets peer review any science opinion pieces? That's interesting you should mention that. I was talking about this with Paul Offit this morning when we were recording Beyond the Noise. I don't know what the process is, but I think it's just an in-house editorial board. And I doubt they have any science expertise. Should they peer review it? It needs to get some kind of scientific review. Because if there had been some scientific review of the Chan opinion piece, it would never have been published like that. She would never have been allowed to say all the things that she said, which were just blatantly misleading and false. Like the vast majority of evidence indicates it came from a lab. This is just wrong. Wrong, wrong, wrong. Why do you let her publish that, New York Times? Because you don't know any better yourself. So, yeah, I think there should be some kind of review. How that's constituted, I have no idea. And if you can't figure it out, don't publish the pieces. What's the purpose of saying, I think it came from a lab? Who does that serve? Right? I don't get it. Artemis says one reason why it's so hard to use data to convince the conspiracists is that they are wrong, is that data is rational and conspiracy is irrational. So we are in two very different universes. That's correct. So I got an email the other day from a guy who said, Racaniello, every point you made in your rebut of Chan is wrong. You didn't add anything to the conversation. And I'm thinking, what part of the data don't you get? Right? You obviously don't want to get any of it. So you're right. It's irrational thinking. Totally. Hmm. Can't we make vaccines for birds which are pellets of food? Well, you want to control the dose, right? Because I just don't know how that would work. You know, if you put there's a big tray of pellets, right? And the chickens all run for it, and some get a lot and some get not. I don't think that's the way to go. And I don't know anything about immunizing animals, right? But that's my opinion there. The food chain in the first world is a marvel of efficiency. They vaccinate cattle, etc. If you'll agree to pay more for your hamburgers. I don't know anything about it, but I'd love to learn. Artemis says pig vaccination, assuming we have a working vaccine, is expensive, but having to kill millions of animals who got or might get sick may cost even more. Yeah, I don't know what the trade-off is. You know, in Malaysia in 1990, whatever it was, the outbreak of Nipah on a pig farm, they culled 1 million pigs. Very costly. Yep. Vista says vaccinating cattle or pigs against H5 if transmission continues. Is there any benefit towards stopping mammal adaptation? Remember, the vaccine is not going to stop infection. It will prevent disease. And so it's not clear that it's going to stop adaptation at all, right? Influenza viruses still circulate extensively in humans despite vaccination. So that wouldn't be the goal for animals. It would be to keep them healthy so they can be brought to market. This is a question from Mark Martin. Are you still here, Mark? You may have left, but for the pneumonia vaccine, what's the difference between the polysaccharide and the conjugate? I don't know. So they're different, right? Yep. The polysaccharide, which, as you could guess, is comprised of polysaccharides, has 23 serotypes. The conjugates are made by attaching the polysaccharide to a carrier protein, which is diphtheria toxoid, which increases immunogenicity of the antigen. And so the polysaccharide vaccines are not conjugated, so they have lower immunogenicity. All right. Did I read correctly that Finland is vaccinating the public against H5N1? I don't know. I didn't hear that. Yeah, here it is. June 25th. Finland says it plans to begin vaccinating vulnerable populations like farm workers against bird flu as early as next week. They bought a vaccine from a company. Finland to roll out vaccines for bird flu as human cases of virus spread across the globe. So you're right. Is that jumping the gun? Okay. In 1976, there are a couple of cases of influenza at Fort Dix, New Jersey. Some scientists believed that it was going to be a pandemic, so they made a vaccine against that strain and vaccinated 140 million people. The virus never spread, and the people got Guillain-Barré. There were hundreds of cases of Guillain-Barré. So, my opinion would be wait, because there's no evidence that there's an outbreak anywhere. If you've got the vaccine, as soon as you see case numbers rising, then you can immunize. You can catch it pretty quickly, but you risk other issues. And you know that made people very unhappy about vaccination back in 1976. Do you remember that? Gerald Ford was president. He ordered that we vaccinate people against 1976 swine influenza. Pete writes, up to 2 non-party appointments can be made to UK House of Lords this month. Alexandra Freeman applied to advise on risk and evidence in science to the politicians. Okay. I hope Alexandra Freeman gets it right, because it's great to have a scientist explaining things, but if you explain the wrong things, like the alpha is more transmissible, you're not doing anybody any good. I'm not saying that Alexandra did that, but you understand my hesitation. Matters Microbial talked about some politicians that have doctors and scientists on their staff because they want to be informed and see lack of knowledge as embarrassing. I don't know how that works. It must be a minority of politicians. And I mean, how do we know the scientists and doctors are knowing anything? Look, a huge number of doctors in this country don't give Paxlovid. They don't think it works. They don't think it's good for you. What the hell is this all about? Just talk to Daniel Griffin. I don't know what's going on. Congressional Fellows Program. Yeah, I know about that, Mark. What has it done? Why is the House investigating the origin of SARS-CoV-2? Why don't they tell them, folks, it didn't come from a lab? Obviously, it doesn't work. Why is he still head of the USPS? It's a lifetime appointment. RE Alternative Media have repeatedly told Matt Taibbi and Glenn Greenwald to contact you every time they mention the lab leak crap. No, they never have. Of course not. Of course not. It's too long. We've told them to watch every twiv. They're not going to watch. They'll stop listening after 5 minutes. Will they give you a science Nobel? Never. I will never have a Nobel Prize. It's fine. I don't need one. But I did get the 2022 Richard Ernst Award, which for me is better because it's for science communication. That's great because there's no Nobel for science communication. And this award I was very proud of. Still am. Oh, look at this question. If you were going back to lab work, what would you be working on? Good question. You know, I can't work on poliovirus anymore because they're shutting down poliovirus research by 2025. So get this. Poliovirus continues to circulate. You know, we've reduced the number of polio cases globally, which is really good, but there's still lots of cases. The virus continues to circulate. Yet they're going to stop researchers from working on the virus next year because they don't want it to go from a lab to the environment. So let me understand this. It's already in the environment and you don't want it to go from a lab to the environment? I don't understand this containment garbage. If I could, I'd work on poliovirus because a lot of things were not finished. But if I couldn't do that, what would I work on? I would not work on coronaviruses, although the smart money would say to work on it because it could get funded, especially if I wanted to do some wildlife sampling, right? Go out and sample some bats. Well, it would certainly be some kind of a virus, and it would have to be an RNA virus. Maybe influenza virus, which is what I started as a PhD student. Yeah. Thank you, Jeff. Everybody should bring up twiv in comments about every story about a virus or virology. Yep, I agree. Just hammer it home completely, incessantly, until everybody is frickin' listening. And you know, we need to have shorter form, I agree, because it's a bit daunting to people. You know, I broke out the SARS-CoV-2 origins thing on its own, and it was still 55 minutes. It's too long. It needs to be much shorter. But yeah, I want to have places where people can go and listen to something that explains why this isn't so or is so. Because right now, the twiv is kind of an amorphous thing, right? It's a conversation that kind of wanders. You know, if you want to learn something, it may not be the best format, right? But if you can go somewhere and say, this video will tell you where SARS-CoV-2 came from. This video will tell you why mRNA vaccines do not cause cancer, right? That would be a good library of stuff to have. So we'll make that one of our goals, to make that kind of video library. Klaus thinks a conversation between me and Osterholm would be like a Newton-Leibniz. I don't know. I don't think he likes me very much. I invited him to twiv years ago, and he said no. Can you comment on an article in Cell.com regarding multi-dose BCG as preventative for COVID? So I haven't read that article, but you know, what's going on here is a general activation of the immune response. You can have you can get infectious attenuated vaccines like measles, mumps, rubella, or oral polio vaccine, and they will give you a couple of months protection against SARS-CoV-2. Well, that's the idea anyway, because it works for other respiratory viruses. Nobody's tested it for SARS-CoV-2. It really needs a clinical trial. And I suspect BCG is working in the same way. But I don't, it's transient. Just get vaccinated. Get vaccinated. The vaccines work. Andrew really hates how some people are outright against virus surveillance now and claim we are safe for not knowing because scientists are irresponsible. Yeah, this is ridiculous. Everything we know about what's out there comes from surveillance. There haven't been any accidents leading to big problems. Why the hell do you have to change things? Because you got some bee in your bonnet that says it's a problem. Who cares what you think? Who cares? It's not up to you. Let the scientists do what they need to do. They can do things safely. Oh, Vincent, you're conflicted. Why am I conflicted? I don't do any science anymore. I'm just telling you my opinion from watching virology for 45 years. And we are not irresponsible. We are really careful about what we do. The irresponsible comes from people who have no idea how virologists work. So don't listen to them. Come to think of it, if you want to know something about virology, you should ask a virologist. Try something novel instead of a lawyer or a politician or an economist or Jeffrey Sachs. Right? What the hell does he know about any of this? But there is no value in expertise. Did you know that? There's no value in expertise. Racaniello spent 45 years working on viruses, but he doesn't know anything that I don't know. Okay, let's talk about that. So next time you fly an airplane, are you going to let a surgeon fly the frickin' plane for you? The next time you have surgery, you're going to let an airline pilot work on you? What are you, out of your mind, folks? So dumb, ignorant. Oh my God. HPV vaccine hasn't been tested on older people is an excellent reason for not giving it to older people. Brings that question of why it hasn't been done. I don't know. You have to ask the drug companies who make it. Mainly, Merck is a big one, right? Why don't they test it in older people? I don't know. Maybe it's just a small market. Bryce writes in the excellent twiv rebuttal to Chan's New York Times opinion piece, there was a brief mention of a paper that shows a single lineage based on new early sequences. Could you cover this paper or its flaws? Yeah, well, if you'd like, we can do that. Yeah, it's flawed. There isn't a single early lineage. There were two, but we could do that, sure. Laura writes, I think you have to chalk it up to being an unfortunate coincidence. The lab was in Wuhan. Political bullies attack anything new or different. It's a sign of brain damage because thinking hurts. Yes, thinking does hurt. There are 4 labs in Beijing that work on coronaviruses. So if the pandemic had started there, they would have blamed those laboratories. And that's one of the pieces of evidence that is cited for a lab leak. There's a lab in Wuhan that works on coronaviruses. How could it be coincidence, right? Well, yeah, that's how coincidence works, If a person is looking for information, TikTok is looking in the wrong place. Well, can't we change that? Can't we make, we have a channel, we have a microbe TV channel on TikTok. It doesn't have 0 views, right? It has maybe a few thousand, not huge, but I think we could change it. If you give up, then all is lost already, right? Yes, and this is a good point. People waste hours on Joe Rogan's long form interview of celebrities, and then they claim that twiv is just too long. You're absolutely right. 3, 4 hours, not a problem. Well, they'll watch a football or a baseball or whatever sport game for 3, 4 hours. And I understand that's their prerogative. They can do whatever they want with their life. but do not claim ignorance because you refuse to use your brain properly. Don't go over to the conspiracy side without using your brain. They're just not going to do it. Lise writes, the scientists and students at OSU that I send the long form episodes to really like it. Please keep it. We will. No question about that. Clinical immunology, tracking onset and course of respiratory virus infections. My suggestion for an office hours. I'd like to get more guests here on office hours. It's a nice format for a guest because not only can I ask them questions, but you can participate as well. So my goal for the summer is to try and get more guests, not just the usual people who come here, who are wonderful, but some different guests that I've had not had on any of the other programs. Yeah. John writes, if anyone wants to hear my first very amateur differential after this, check out the latest TWIP 237, around 6.5 minutes. I got it right. Thanks to the parasitology course textbooks and episodes. Oh, look at this. James writes, an Aussie doctor does back to the science that is 100% John Campbell refutations. Why do we have to have this? Why? Because John Campbell got wealthy on YouTube by spreading misinformation. So now we have to refute that. We have to spend time refuting it. So I presume, James, that this doc is going to take care of the mRNA vaccines causing cancer, so I won't need to do that. Have you heard of Daniel Lucy? What do you think of his idea that was brought into the market before it came out? Yeah, it was brought into the market by animals, for sure. How would it be brought into the market by a human? Where would that human have got it? Oh, from a lab? Sorry. There's no evidence that it came from a lab. Where is the epicenter then where that infection began? Because remember, there are two lineages at the market. So they both originated there. So they were both, that likelihood that both were brought in this way is vanishingly small. So this doesn't work at all. He's wrong. And many people say, oh, it was an amplifier. No, it's not an amplifier. It's the only place in Wuhan where you see early clustering of cases and two different lineages. All the early cases are accounted for in the market or in the vicinity of the market. It is really, really good epidemiology. See, people realize that this data are devastating to their hypothesis, so they try and trash it by saying nonsense like this. Do you anticipate more antipathy with the possible economic damage with swine bird flu? What do you mean, in general? Like antipathy towards me, antipathy towards science? I don't know exactly what you're asking. Why don't you restate, please? All right, here's an interesting thought. All media need to do is require degrees for certain journalists, such as science reporters. HR would filter those out. It would be better than what they do now. I teach a lecture, a single class to science journalism students at Columbia every year. These are students who are going to get a master's in science writing and journalism. Some years, they have no idea what I'm talking about if I talk about DNA, protein, mRNA, nucleotides. They have zero science background. All they are learning is how to interview people and build a story around the interview. Now, the professor's great, Jonathan Weiner. He knows his science, but you can't teach these students science. We tried it, actually. I tried one year to give so I give a virus lecture, a 3-hour virus lecture every year. And he said, let's try teaching them some basic biochemistry first. Boy, they just fell asleep. They totally had no idea what was going on. They didn't have the background to handle it. They could deal with the virus stuff. So I'm not convinced that getting a degree in science writing is particularly useful. It doesn't really help you cut through the nuance that science is. Now, I'm not saying that every journalist needs to be a scientist. What do we do though? I don't know. Journalism schools should also add science to their course load. I don't think they have time. I know many of these students are not scientists. They never had any background in science, but it could get them a job. So this is the attraction. Hmm. There's a good podcast called This Podcast Will Kill You with two NPs who did a whole pod on the different forms of vaccination and explained in detail conjugate vaccines. Good. Just check that out. I know them. The investigation, I don't know which one we're down to talking here, is being held by those politicians who aren't embarrassed by misinformation. Apparently not. Logotic says, I recall the 76 swine flu scare. When Chevy Chase portrayed Gerald Ford on Saturday Night Live, he always had a syringe sticking out of his arm. It's perfect. Oh my gosh. Yep. Mark Martin says conspiracies are weirdly attractive to some people. My late brother believed in the Roswell UFO landing. I could never get him to be reasonable. Very smart fellow. Miss him terribly. Yeah, I don't believe in any conspiracy theories. No, there's facts, there's data, and you put it together. That's all there is to it. And if you don't have all the data, you wait. Here we have some information on the Postmaster General can be hired and fired only by the Post Office Board of Governors. So far, they don't want to. They are allowing him to dismantle the U.S. Postal Service. He's such a slime ball. I was watching this hearing in a small town somewhere. People were saying, we can't, the hours are cut. We can't get to our post office, blah, blah, blah. What's going on? He says, I don't know. I'll look into it. Obviously, they're just cutting resources so that the whole system falls apart. Ay, ay, ay. Here we go. Conjugate pneumonia vaccines uses T cells and make memory cells. Polysaccharide only uses B cells. There you go. This is all about the USPS, so I brought up something sensitive here. Do you know what would teach MAGA a lesson about privatizing the USPS? The high cost of first-class postage charged by FedEx, UPS, DHL would serve them right wing. Yeah, but they're owning the bloody company, so they get the profits. Good night, Mark Martin. Enjoy your dinner. It is 9:41. I think I'm going to skip the mini lecture for tonight. We'll just wrap to the end of the questions here and say good night. Have Sabin polioviruses used for oil vaccination temperature sensitivity? Yes, they have They are temperature sensitive. They have to be grown at lower temperatures. Yes. Susan, a lawyer and political commentator, said on YouTube that in view of the rise of bird flu, maybe we should lay off milk for a while. I commented that he should watch you and Daniel Griffin. Well, I always tell Daniel, humans don't need milk. There's no reason to have cows, right? We got mothers that can breastfeed for a while, and then you can supply your nutrient needs through other foods. We don't need milk. Milk is an industry that has been put together to give people jobs and so forth, and that's fine. But you don't need to drink a glass of milk a day. Maybe we should lay off milk a while. You can't just suspend the whole industry. You got a lot of people working. You have farms operating. You have the animals. You can't just turn everything off. Once you're in this problem, that's it. Tom writes, what is truly irresponsible is misinformation, especially that promulgated by a credentialed renegade authority, such as age denialists, anti-vaccine doctors, climate deniers, and that ilk. Yes. But I would argue that some of the writing in the Chen piece is also misinformation. It's very subtly couched, but it is misinformation. But yes, people who say that vaccines are all bad and never been tested, like RFK Jr., this is irresponsible. This is costing people's lives, and that is just wrong. Totally agree with that. Same thing with climate change deniers and so forth. They deny it for their own economic benefit. I was in Sweden, I was in Finland. People said, Vincent, we look up to the US, but what are you guys doing these days? Good question. Hello, Veronika. Privyet. Heat wave in Moscow. Any progress? Angela's working on it, yes. I think her goal is to get a couple of episodes in the can. She's recorded one, so should be out sometime this summer. Yep, for sure. Jim Murphy writes, hey, Vincent, don't stop what you're doing. This is great, valuable, necessary work you pull off at the people at twiv. Michael Osterholm, Debunk the Funk, Dr. Wilson Susan Oliver are doing essential science education. I need you to keep it going. Thanks for your efforts. Nice job with the broken comments. Thank you very much, Jim. We will keep going. We made this company. It's working. It's going to go on. Nobody's going to stop us. However, I'm getting worried about litigation. So we're starting to protect ourselves, sadly. If you notice now. And I should say, information on our programs should not be construed as medical advice. If you're sick, see a doctor. So that's now in every video. Libertarians consider scientists to be a problem as they interfere with industrious businessmen. They don't like regulation, preferring the market, that liberty and freedom nonsense. Some things are not consistent with freedom. Public health does not work if you give people choices. Public health works when people do what you tell them to do. The assumption is the people making public health policy know what they're talking about, and everyone falls in line. It's like I pick a doctor, and I'm in your hands. I picked you. Whatever happens, it's my choice that I made. But you're the boss. You know what you're doing. And it's the same with public health. Now, people have lost confidence in public health because there have been missteps. And you can list them like crazy, all the missteps that CDC has made, right? Screwing up the diagnostic tests for COVID, masking, confusion about vaccines. They need to get their act together. And gaining back that trust is hard, but we should be able to trust them. In Sweden, if they say no more pap smears, that's it. People say these people know what they're talking about. They're the experts. We're going to listen to what they say. Okay. He says it came from another wet market. Why? Why does it have to come from another wet market? All the evidence is that it's spilled over there, including environmental evidence, right? This is dumb because, okay, if it came from another market, where is that market? We have samples from this market with both lineages, environmental samples that also have mammal nucleic acid in them. So it's clear that it's the epicenter. If it started somewhere else, that other place would have been the epicenter, and that would show the epidemiological footprint. So no, this is completely untenable. It did not start somewhere else. It started at the Huanan seafood market. Read the paper, Lucy. Oh, Abdul Rahman. Antipathy towards the scientific community. A big part of the whole unpasteurized trend is due to possible damage done to flocks now that avian flu was found in beef. I think most people support science and medicine. I think the people who don't are a minority, but they they really color it for everyone else. They make it look like nobody believes in science. So I think it may change slightly, but I don't think it's ever going to be 100%. But I don't know. It's I can't tell. Tom writes, the energy, Brandolini's law, the energy to refute bullshit is an order of magnitude greater than the energy to generate. Corollary, Brandolini sets the minimum only. That's really good. Where is he reading these things on the screen from? Well, folks, you are typing comments in the chat. So if you go to YouTube and you're watching this live stream, on the right-hand side, there's a chat where you can type in comments. The software I use for streaming displays those chat comments in a little column. And if I click on them, they disappear or they appear. It's a very nice implementation of the interface. So here, for example, Frank, I click on Frank and here he goes. He pops up and I can make you part of the stream. So the chat always exists to the right, but I make it part of the video by clicking on it. We need more Alan Doves as journalists. Amen. So Alan was trained, he was a PhD, trained in my lab. He got good virology training. Yeah, that's a good way to use a PhD. I agree. In fact, there should be a PhD journalism program. Jeff writes, I had no idea such a program existed, the Masters in Science Writing. However, I think requiring them to have an AS would be better, or a BS. I don't know what the requirements are to get into that program. I assume it's some kind of bachelor's degree, but not necessarily a science degree. Yeah. Journalists are competing for fewer spots where they are expected to do more with less editorial oversight and fact checking while being pressured to cut costs by private equity. Yes, there's a lot of pressure. I understand that. And they have to produce more all the time with less checking and less resources. Totally agree with that. But it's not an excuse to produce poor stuff that misrepresents the field. So many poor articles about COVID during the pandemic. It was unbelievable. That's where twiv comes in. We give you a really good analysis of the science, but how to fix it, I just don't know. It's very hard. It's pathetic that media never required a degree for certain reporters. I hear some talk like they get educated enough from interviewing. Well, I think the approach is that you teach people how to interview. You teach them how to ask the right questions and then take the answers and formulate an article. And then you talk to more than one person, ideally, when you're looking at an issue. One of the problems I see is that what do you do if people have differing viewpoints? Like if you ask someone, is alpha more transmissible? One person says yes and the other no. They seem to just decide on yes because it makes a sexier story. That's what I fault the New York Times for doing all the time. No. Sometimes the 1 out of 10 is right. So how do you get that in journalism of science? How do you figure that out? The mainstream idea may be just wrong. I think would be more interesting to figure that out than to just publish what everybody's saying and call it a day. But nope, they don't seem to want to do that. The milk industry is a byproduct of the veal industry. Okay, I can buy that. Yeah. All right. Now people are saying why they need milk for my latte, cheese, pie crust, ice cream, pie fillings. Well, you don't need to drink a glass of milk a day. That's all I have to say. I'm not going to get into the cooking argument because I understand what you're saying, but you don't need to drink a glass of milk a day. Maybe the industry could be made much smaller. Visto, hai visitato in Italia durante il tuo viaggio? Sì, ho visitato in Italia. See, okay, let's wrap this up. Milk cows have short lives, deliver 100 pounds until they can't, and then sent to slaughter. And their male calves are created for veal. I'm a veg. I love starting topics. I started U.S. Postal Service and I started milk. Did you know they sell cultures to turn soy milk into soy yogurt? They might have something similar for soy cheese, but I don't know. I agree that milk products are great, but you don't need to drink a glass of milk a day. Jill, it's late to the party, but I want to say thank you to Vincent for fighting the good fight. It must feel Sisyphean, but you and the rest of the Micro TV gang really do make a difference. Thank you so much. We really appreciate your support in words. Cheese is essential. Do goats get H5N1? I don't know. It's a good question. Let's look it up. Yes, the University of Minnesota Veterinary Diagnostic Lab found a positive goat for H5N1. So yes, they can get H5N1. Why do people like to take his advice? He's not a virologist, okay? Doesn't know anything about vaccines. Oh my gosh. Yep. Goats reported to have H5N1. We got that. Hello, Doreen. Nice to see you tonight. Welcome. Let's see if we have any more questions here. You guys are talking about food. Masking. I once heard someone say you need to wear a mask only when talking. No transmission if silent. Luckily, I was wearing an N95 so they couldn't see me laughing. Of course, just breathing is enough to transmit. It's too late for the journalism majors who may have avoided the sciences from as early as middle school. We need to start in grade school, teaching logic, history, and scientific method to anyone. I totally agree. Johnny said, can we use viruses to target cancer cells in vitro and change their DNA back to healthy cells with Cas9? No, but we can use viruses to kill cancer cells in vivo. And I was going to actually talk about that tonight in the mini lecture, but we didn't get around to it. So another time, come back and we'll talk about that. Hi, Vincent. I've recently started following twiv. Really enjoy it for COVID. And people now, do we need to take any precautions? As there still seem to be a minority ending up in a hospital. So the ones ending up in the hospital are largely 65, 70 and older, and with other health issues, comorbidities. But for the vast majority of the population, the level of immunity is very good. And under that age, the illness is mild, especially if you've been vaccinated. And even though there's this variation in antibody binding epitopes, the T cells seem to be taking care of disease very well. More and more evidence to that effect. Yep. Great point about not demanding evidence and follow up. No article I've ever read discussed what transmissibility is. No, they don't know what it is. I mean, Ron Fouchier said to me from the start, this idea that the variants are more transmissible is absolutely insane. There is zero evidence for more transmissibility. It's being all mixed up with spread of a virus in a population of humans that are going back out into the world after being locked down. This is just a conflagration of bad epidemiology and virology. Why am I wearing the ass cap? Well, the cap is kind of a joke, right? Right? It's a cap, ass cap, so that means that I'm an ass. It's a joke. I was wearing it to protect my head from the sun, and it was the hat I found. All right. Time to wrap up here, folks. It was great to see you all tonight. Oh, by the way, Vanity just showed up late, but thank you for joining us, Vanity. Hope your friend is okay. Thanks to our moderators tonight, Vanity Nutrition, Tom Steinberg, Les Fabi, who was here earlier. One more person early on. Stephanie. Stephanie was here. Tom, did I get Tom? Thank you all for moderating, and thank you all for coming here, all of you. We did pretty well on the likes. We appreciate it. Next office hours will be a week from today. That is Wednesday, July 3rd. I don't expect many people to come. It's the day before the 4th of July, but I will be here. Need to get back into the habit of doing a weekly office hours, whether or not I have a guest. I don't know. I'll do my best. But anyway, folks, thanks for coming. Hope you learned something. Just be critical when you read things about science, and that will help, and know who to ask. Until next week, please be safe. Good night, folks.